So I'm on live now. Oh, you guys are here. Sorry, I had to step away for a while. Okay, so you're signing in. I see 1, 2, 3. Okay, 17 people signed in. 18. It's now 10:29, so I'll give it 1 more minute and just wait for the others to get here. Hello, Heaven. Hi, Hans. Okay, it's 10:30. I'm gonna wait 1 more minute for those who didn't sign in as yet. Oh, Noel, I forgot to email. I don't know if you got my email, but your benzaldehyde is in my office. I think I emailed you, so make sure you get that, okay? Remember to identify yourself. Someone said, hi, Dr. Murray. I don't know who it is. So remember to identify yourself. Okay, I think that's everyone now. Yeah. All right, just one announcement. So today after class, you would need to go to the lab and get your cells growing for tomorrow's lab. So tomorrow you will be extracting the plasmid. You'll be extracting the plasmid that you transformed into your cells. But in order to have enough plasmid to do the extraction, you have to allow the bacteria that contains the plasmid to grow. As the bacteria grows, it replicates the plasmid, so you'd have enough plasmid for tomorrow's extraction. So to do that, you will follow the instructions on your lab protocol that says to set this up a day before lab, Wednesday before lab, which is you will take a colony from your plates, from the plates which you will find in the refrigerator in room 240, use a toothpick and remove 1 colony and put it into one of the test tubes. Brandon is going to have the test tubes ready for you upstairs in 240, the test tubes with the toothpicks. Okay, so make sure you have that, make sure you inoculate your cultures, okay? Someone is saying they can't see me. Let's see, who is that? Someone said, I can't tell if I sign in, can you see my submission? You have to include your name. If you don't have your name, then I don't know who you are. Okay, so make sure you have your name there. Your cultures, Rayford, your cultures are in the refrigerator. They're in the refrigerator in 240. So make sure, so again, you'll go upstairs, find your plates that have your transformants, take a toothpick, you will take one colony from the plate, and then you take that, you scrape the colony from the plate with the toothpick, and then you take that toothpick and you inoculate your liquid culture. I want you to do 2. Each group should do 2 cultures. But tomorrow you'll only be extracting from 1, but I'm asking you to do 2 just in case there's a problem with growth. So do 2. Yes, you could do this up until 3 p.m. Brandon says he has lab, I think, some other plans he has. So you have up until 3 p.m. to do this. Charlotte, yes, I am receiving your responses. Okay, are there any other questions before I start? If you have any questions before I start lecturing, let me know. Because of the delay, I have to wait. So I'm just waiting for questions. Okay, looks like nothing is coming in right now, so we'll get started. So, oh, Talissa, no free time before 3. We should be finishing early in today, so you could head upstairs actually right after this class. Okay. Oh, can you get your test back? Ivan has the test. He should have them ready today. So you could, I will send him an email and ask him about getting that back to you. Okay. Can I see your responses? I am seeing responses, but you have to include your name. If you don't put your name, I don't know who you are. So I'm just seeing a question. Can you see my response? You have to let me know who you are. Yes, there will be a TA up there after class to help you set up the lab. Brendan is going to be available to help you set up the lab. If no one is there, email me and I will contact the TAs to make sure someone is there, but Brendan is supposed to be there. At least he will have the supplies out, so you could do that. Okay, I think we should get started. No more questions, let's get started. I don't see any more. Okay, so one last time, any more questions before I get started? All right, so let's go. So we're talking about the genome, the bacterial genome. When we talked about the bacterial chromosome, we said that the bacteria contains one circular double-stranded chromosome. And we looked at some of the features on the chromosome. We mentioned that on each strand, you have genes that are transcribed, which is unlike what we see in the eukaryotic genome. So we have genes that are transcribed on both strands, and they're transcribed in opposite directions. We also looked at the transposable elements. That's the insertion sequences, the IS elements, and the composite insertion sequences, which contain two insertion sequences that bound antibiotic resistant gene. So we saw that. And then we saw that bacteria contain extra chromosomal material in the form of plasmids. And plasmids, we learned in chapter 9, can be used for molecular cloning. Okay, so on Monday, when we ended, I asked you guys a question, and I don't know if anyone gave it thought. Let's see how much thought you gave it. So we were talking about the three methods of horizontal gene transfer. Could anyone tell me what those three methods of horizontal gene transfer are? If you give the answer, again, put your name there. No one? Can you give me one? One method of horizontal gene transfer. You did one in lab. What was that? Okay, so no one is answering. Okay, let's move on then. Thank you, Warren. Transformation, conjugation, transduction. Great. Okay. All right. I got it. Got the answers. Great. Okay, so we talked about transformation last time, and I asked you guys a question, and let me pull that slide up. This is really awkward with this delay. It really is awkward. Again, I got a question. Can you see my responses? I don't know who you are unless you put your name there. Okay. All right. I don't know. When you say what I answered, I don't know who you are. You have to put your name there. Yes, Temitope, I see your responses. Isaac, Abraham, Linnea, Nicholas, Ezra, Hans, Abraham, Zach. So yes, Rayford, I am seeing the responses. Don't worry about it. I'm seeing the responses. Okay? You just have to identify yourselves. Okay, so here was the question. Let's pull that up. We're talking about the difference between selecting and screening. And let me see where we are here. Okay, let's pull this slide up here. All right, so we're talking about the difference between selecting and screening. And we said, given the example here, you're seeing a donor cell that is wild type for the histidine marker and a TRP marker, and it's being transferred, or it's donating its DNA to a double oxotroph. And in order to select for TRP plus transformants, you will plate on minimal media that contains no histidine and no tryptophan. And if you had to do the reverse, which was to select for His transformants, you would do the opposite. You would plate on minimal media with tryptophan and no histidine. The last question I asked was, how would you screen for co-transformants? That is, transformants that are both histidine and tryptophan plus. So you did the transformation, you know how to screen for one marker. What if you had to screen for both markers? What would you do? How would you go about doing that? Again, if you answer the question, you can't see the slide. Oh, I'm sorry. Sorry. I'll get the slide. Okay, let me know if you're seeing the slide. Oh, so Nicholas, you said with electrostatic screen preventing, could use the FB chat group to ask questions instead. Yeah, we'll rethink this, Dylan. We can't do that right now. Okay, I'll just give the answer. Okay, so I think it's becoming a little awkward here to ask questions. So here are here's the answer. What you would do, you will screen for co-transformants. That's a strain that is both histidine and tryptophan plus. What you would do, you will test your TRP transformants, for example. You will test those for the presence of the His marker. So if you have a strain that has the histidine marker, has the histidine gene, has the tryptophan gene, you know it was transformed with the tryptophan marker. What you will do is take those tryptophan positive cells and you plate it on media that lacks histidine. And so that way you could screen for both at the same time, okay? So that was transformation. The second method the second method of vertical transfer is conjugation. And this was first demonstrated by Lederberg and Tatum in the 1940s. And if you look at this slide, you'll see two strains A and strains B. In strain A, you're seeing that this strain is oxotrophic for two markers, that's the methionine and the biotin, but it's prototrophic for threonine, leucine, and thiamine. Strain B is the reciprocal of that. So strain B is oxotrophic for threonine, thiamine, and leucine, but prototrophic for methionine and biotin. But you'll see that strain A, when placed on minimal media, there's no growth. Okay? Strain B on minimal media, again, there's no growth. But when you mix them, they are able to grow. That mixture is able to grow on minimal media, suggesting that somehow, once they're in contact with each other, something is happening that allows them to grow. So let's see what is happening. Well, it was concluded that what was happening was conjugation, that there's some direct contact between these two strains and they're able to exchange, transfer their genetic material. Okay, I just saw Dylan's answer. Plate with histidine, then take cultures and grow it and plate. Great, Dylan answered the question. Great. Nicholas, you're asking if the slides will be posted. Yes, they will be posted. They're on the Learning Hub, automatically on the Learning Hub. Oh, the slides, yes, I will post the slides as normal, as usual, on the Learning Hub. And this video presentation is going to be on the Learning Hub also. All right, so let me switch out of this. 1 second here. Alrighty. Okay, so this is conjugation. So bacterial conjugation requires direct cell-cell contact, okay? So in the previous example here, we have strain A and strain B. In order for conjugation to occur, these cells must come in contact with each other. Unlike transformation, there's no direct cell-cell contact that is needed. In transformation, the competent cell has to just be exposed to DNA in the environment. But with conjugation, you must have direct cell contact. And so this is a pretty nice EM figure showing you that. So not all bacteria are able to initiate conjugation. In order for a bacterial cell to initiate conjugation, it must carry a special type of plasmid known as an F plasmid. An F plasmid is a conjugation plasmid. So cells that contain this F plasmid are said to be F plus. F plus cells are cells that have an F plasmid, and cells that have an F plasmid are able to initiate conjugation because they contain some of the genes necessary to create the structures needed for conjugation to occur. So we are seeing here in this slide a bacterium that has a bacterial chromosome, like all bacteria, bacterial cells, but it also has a special plasmid, that's the F plasmid, that allows for conjugation to occur. It helps to initiate conjugation. So cells that contain this plasmid are called F plus cells, and cells that are lacking this plasmid are called F minus cells. F plus cells are able to initiate conjugation because they create what is known as an F pilus. That F pilus extends from the cell wall of the F plasmid, and it attaches to, it extends from the cell wall of the F plus cell, and it attaches to the F minus cell and creates a bridge between those two cells. Once that bridge is created, then the pilus, that bridge begins to contract and pulls the recipient cell close to the F plus. So it pulls the recipient cell close by. And once the cells are in close proximity to each other, then the F plasmid is transferred, and you can see that here. But what is being transferred is only one strand. Only one strand of the plasmid gets transferred. So one strand of the plasmid moves over into the F minus cell, and what you end up with are two F plus cells. Okay, so one strand gets transferred. As the strand is being transferred, it then replicates. So you end up with a double-stranded plasmid in the F minus cell, and the F plus cell, after losing that one strand, the remaining strand replicates. So you end up with two F plus cells. So a mating, so conjugation is also known as bacterial mating. Okay, so when you have a mating between an F plus cell and an F minus cell, the final products are two F plus cells. So a mating between an F plus and an F minus will produce two F plus cells. Okay, I'm gonna check the chat to see if anything is happening here. Okay, let's move on then. Well, let me pause. Are there any questions so far? Okay, so let's move on. Okay, sometimes after the transfer of the F plasmid into the F minus cell, most of the times the F plasmid will remain outside of the chromosome. But on some rare occasions, this F plasmid could integrate into the bacterial chromosome. So looking at this slide here, we could have this F plasmid integrating into the bacterial chromosome. And it integrates at the insertion sequence sites. If you recall, we said that one of the elements you'll find on the bacterial chromosome are these insertion sequences. Well, you also have insertion sequences on the F plasmid, and because those sequences are similar, it's a place where recombination would occur. So you could have recombination occurring between insertion sequences on the plasmid and the insertion sequences on the chromosome, and that will cause the F plasmid to integrate into the genome. When an F plasmid integrates into the genome, you create what is known as an HFR cell. HFR stands for high frequency recombination cell. So it integrates into the genome here. Okay. Once integration occurs, there are times where after the F plasmid integrates into the genome, creating an HFR cell, this portion of the genome that contains this integrated F plasmid could then transfer into another cell. So you could have, basically I'm saying, you could have a mating between an HFR cell and an F minus cell, right? Because an HFR cell really is, it's just a cell that contains an F plasmid integrated into the genome. So Kiara, your question is conjugation an example of horizontal transfer? Yes, it is. It's a example of horizontal transfer. So transformation is an example of horizontal transfer, and also conjugation is an example of horizontal transfer. And the third one we'll talk about today, transduction, also an example of horizontal transfer. Okay. So let's go back here. Okay, so we have the F plasmid integrating into the genome, creating an HFR cell, and this HFR cell could initiate conjugation with an F minus strain. Okay, initiate conjugation with an F minus strain. Okay. Depending on where that plasmid inserts into the genome, you could create different types of HFR cells. So here we're seeing a bacterial genome. It's showing you 3 regions, A, B, and C. And here is your F plasmid inserting and creating different HFR strains. So if it inserts between A and C, that's one type of HFR strain. If it inserts between A and B, here's another type, another type, another type. And then also the orientation the orientation of insertion also is another way of differentiating the HFR cells. Because if you look at this one, so the orientation of the transfer is this way, okay? And here it's this way. So these are different. These will all be considered different HFR strains. So it says here, an F plasmid can integrate into the bacterial genome. When it does so, it's called an episome. So an F plasmid in the genome is called an episome. And it says HFR strains differ in the location and orientation of the integrated episomes. And like I mentioned before, these HFR strains can initiate conjugation and donate their portions of the HFR plasmid into an F- strain. So here's an example of this donation between, or mating, I would say, between an HFR donor strain and an F- recipient strain. So here we have our HFR strain with the integrated plasmid. And because it has the F plasmid, this is the entire F plasmid now in the genome, it is able to initiate conjugation between itself and an F- cell. So here's your F- cell. The pilus is created and it's then contracted, which pulls the F- strain close. And then you have a single strand break, a single strand cut on the F plasmid, that, or the episome that is in the genome. So you have that single strand cut and transfer could begin. So you have a break and one of these strands from the F plasmid could begin transfer. Transfer begins at what is known as the origin of transfer. So the cut occurs around the origin of transfer, and then it's then transferred into the recipient strain. But if you see, as it transfers into the recipient strain, it also takes along some chromosomal DNA. You seeing that here? So some chromosomal DNA is also transferred into the donor strain. And in order for, because this contact between the two cells it's very labile, it's not stable, it doesn't stay together for very long, what happens is that only a portion of the F plasmid gets transferred. You see, a portion remains and just a small portion gets transferred along with some chromosomal DNA. Okay, eventually that break occurs, so you end up with a cell that the recipient cell, although it obtained some F plasmid, it did not obtain the entire F plasmid. So it remains F-. It remains F-, but it also now has chromosomal DNA. Okay? So here we have a mating between an HFR donor and an F- strain. The final products are the original HFR and the recipient remains F-. Okay, so in conjugation between an F+ and an F-, you end up with two F+ cells. But conjugation between HFR and F-, you end up with an HFR plus the F-. So there's no conversion into F+. Okay, I'm checking for questions. So Charlotte, you're saying the video froze on one slide? I'm not sure why, but I will post the slides. Okay, so you will eventually see them all. I don't know how to rectify that at this point. Okay. Conjugation could be used, or should I say conjugation? Genes, I should say, bacterial genes can be mapped using during conjugation by a process known as interrupted mating. And so with interrupted mating, what you're really doing is measuring how long it takes for genes to transfer from donor to recipient. So if you recall, when you did mapping in chapter 5, when we looked at mapping genes in chapter 5, we saw that the distance between genes are given in map units. Okay, are given in map units. But in bacteria, the distance are given in minutes. So it's the bacterial chromosomes. is measured in minutes. How long it takes for one gene to transfer from donor to recipient. Okay, and so this is an interrupted mating experiment here. So you have two strains. They're both grown in non-selective media. So you have an HFR strain and an F- strain. So I'll show you here. This is the donor strain. This is the HFR strain and your F- strain. And this is the HFR is the donor and the recipient is the F-. And you're seeing here the features or the genome of both. So here we have the donor has is sensitive for streptomycin. Streptomycin is an antibiotic. So it's streptomycin sensitive, and it is wild type for threonine. So it's able to make its own threonine. It's resistant to azide, which is a heavy metal. It's resistant to TON. TON is a bacteriophage. It's LAC plus, meaning that it can metabolize lactose, and it's galactose plus, meaning it can metabolize galactose. The recipient is the opposite. Okay, so the recipient is resistant to streptomycin, lacks threonine, the ability to make threonine. It's sensitive to azide, it's sensitive to bacteriophage, and it's unable to metabolize lactose and galactose. So those are the markers of the two strains that are being mixed in this interrupted mating experiment. So these two strains are mixed together, and they're grown in liquid media that is non-selective, meaning it allows everything to grow. It allows them to grow for a while, and then you take that mixture of the two cells and you put it in a blender and you agitate them, and that agitation will cause the cells to separate from each other. And then you take the cells and you plate it on media that contains streptomycin. With that, it allows only the ones that are streptomycin resistant to grow. So only the recipient will grow, okay? The ones that are streptomycin will grow. Everything else will die. So you plate it on media that contains streptomycin, which kills all the donor cells, and it will kill any cell that lacks threonine. So it will select against the F- cells that are not mated. Only the ones that mate will able to grow. Once you do that initial selection, then you replicaplate it on these various media. So here you're replicating these original cells. You're replicating them on media that contains azide. That's the heavy metal. You're replicating on media that contains lactose. So only if the cells have the ability to metabolize lactose, they will grow. You replicate on galactose. Again, only those that have the ability to metabolize galactose will grow. And also you replicate it on media that contains the bacteriophage. That's the TON. Okay, so you then look at the plates and see the amount of cells growing on each, and it gives you an idea of how close the genes are to each other. How can you do that? Let's look at that. So again, remember conjugation, you're looking at the rate of transfer in time. How quickly do cells get transferred from donor to recipient? So this is showing you here that cells that were, let's look at this this axis. It says percentage of cells with HFR genetic markers among the threonine and streptomycin sensitive recombinants. We have here 90% of those are azide resistant. Okay, and we could see just looking at this plate here, most of the cells are azide resistant. Okay, 85% are tonomycin, bacteriophage resistant. And it's showing you the time in which it occurs. Because remember, let's think about the experiment again. We had these two strains, donor and recipient. They were mixed together in a tube. They were allowed to mate, but then you interrupted the mating in this blender. And you interrupt at 1 minute intervals. I don't know if I mentioned that, but you interrupted at 1 minute intervals, and you measure the rate of transfer by looking at which markers were able to move over into the recipient each time you did the interruption. So it's saying there here, at around 8 minutes, 90% of the cells were resistant to azide. Around 10 minutes, 85 were resistant to azide. And look at galactose and lactose. They were transferred at a much later time, closer to about 15 and maybe 11, 18 and 15 minutes. And based on that, you could map the genome of bacteria. So again, the bacterial genome is mapped in minutes. And so here's the map where azide is transferred first, followed by ton, followed by lac, followed by gal. Okay? All right. Wow. So we're saying the slides are not changing. I'm not sure why. I will, okay. So what I will do, I will post the slides before class. I will have to do that. I'm not sure why they're not changing. So I'll go ahead and post for next time we meet. I'll make sure I post the slides before class so you could follow along that way, because this is not working. I'm not sure why it's not, why you're not seeing the slides. It is still stuck on, okay. Wow, okay. Sorry guys, I don't know why that's happening. Okay, let's see how far we could get today. Okay, so I heard from a lot of you, the slides are not changing. I don't know why, so moving forward, I will post the slides before lecture. That way you could follow along a little better, okay? But thanks for letting me know. Let's see how far we can get here. Okay, so take home, what I want you to know that when you have a mating between F plus and F minus, know what the product is. If you have a mating between HFR and F minus, know what the product is. Here's another type of plasmid known as an F prime plasmid. F prime plasmids are created when this F factor excises from the HFR chromosome. So you have your HFR here integrated into the genome. You've seen that before. But there are times where this F factor will pop out of the genome. Okay, and when it pops out of the genome, the new plasmid usually takes with it some of the bacterial chromosome, and we're seeing that here. So you have the genome, the F plasmid, but it's also taken up some of the bacterial chromosome. So again, let's start here. F factor integrated into the chromosome, creating an HFR chromosome. And then if excision occurs, sometimes that excision could take out one of these genes. Here it's taking out the tryptophan gene. So you end up with a plasmid that has some bacterial chromosome and the F plasmid. This type of plasmid is known as an F prime plasmid. F prime plasmid. You could have mating occurring between an F prime cell and one that is F minus. And it shows you that here, the F prime cell containing that F prime plasmid, you could have conjugation occurring, and gene transfer you're seeing here, it's being transferred into the recipient cell, and you create what is known as a merodiploid. Merodiploid is basically a partial diploid. So this new cell is diploid for the tryptophan marker only, so it's called a merodiploid. That's a merodiploid. It's still haploid for galactose and histidine, but it's diploid for the trip marker. So that's an example of what a merodiploid is. Okay, let's see how much time we have. Okay. Okay, so it just changed to the F prime. Okay, so let's move to transduction. Okay, so there are two types of transduction. So in general, what is transduction? Transduction is where genes are transferred. Again, another horizontal transfer. It is transferred from donor to recipient, but it is transferred via a bacteriophage. That is a virus that infects bacteria. So it's transferred via a bacteriophage. And so let's see how that works. So there's what is known as generalized transduction, and the other type is specialized transduction. In generalized transduction, any gene present in the donor cell could be transferred into the recipient. That's generalized transduction. In specialized transduction, only the genes that are adjacent to the integrated viral DNA gets transferred, and we'll talk about that some more. So in specialized transduction, there are specific genes that get transferred. In generalized transduction, any gene could get transferred. So let's see how that works. So let's look at this figure. Well, let me see if I have any questions here. Okay, nothing yet. All right. So here we have a bacterial cell, and it's showing you a gene. It's auxotrophic for a gene labeled C, so it's C+. And here is a bacteriophage, and the phage particle infects the host cell by injecting its DNA into the host. Once the phage DNA gets inside the host, we know that it takes over command of that host cell and forces the cell to produce more viral components. So here we have the components that make up the virus being replicated, including the viral DNA. The viral DNA also gets replicated. So the viral DNA is inserted. It commands the cell to make more viral particles, and these new viral particles are then assembled. But here's another thing that happens. It also digests the host DNA. So the host DNA is broken down into pieces. So in addition to making viral particles or particles for making the virus, in addition to that, you also have the chromosomal DNA of the host being degraded into smaller pieces. So as the new viral particles are assembled, so the viral particles, the components of the viral particles are made, and then they're assembled. As they're assembled, they will incorporate some of the host DNA into this viral particle. So you end up with host DNA in a viral particle. That bacteriophage that contains host DNA could then infect another bacterial cell, a new bacterial cell. And that new bacterial cell will now get DNA from the original donor, and that bacterial DNA could get incorporated into the genome. So you end up with a cell here, the resultant cell, that is now C+. So the DNA moved from this original cell here, this host cell, to the donor at this location. Okay, so DNA moved from donor here, donor, into phage. The phage then took it to the recipient. Okay, and the donor DNA gets incorporated into the genome of the recipient. So this is generalized transduction, because any gene that it gets into the phage head could get transferred into the recipient cell. Let's see if there are any questions. No questions. Okay. All right, so this is generalized transduction. I would end here because we have 1 minute, and specialized transduction takes a while. So tomorrow on Friday, we'll look at how we can use generalized transduction to map genes. So you can map bacterial genes based on using conjugation, interrupted mating experiments, using conjugation. But you could also map by looking at co-transduction frequencies. I'll make sure I post these slides today so that on Friday, hopefully we work out the kinks about with this. And also, one question. Let's see, who is it? Dylan mentioned probably creating an FB page to allow for chatting, because this chatting seems to be not flowing as well. So let me know your opinion on that. Would you like us to use Facebook, create a Facebook page, a private group for just genetics students, and we could use that as a way of chatting? So let me see your responses to that. Thumbs up if you think that's a good idea. Thumbs down, no. And I will wait. Okay, Ezra says yes. Stacy, yes. Facebook. Okay. Someone says they don't have a Facebook account, no name. I don't know who that is. Thumbs up. Okay. For the person who doesn't have a Facebook account, can you just create one just for this purpose? You could delete the account after the end of class. And let's see how that goes, okay? So I am going to create a Facebook page, and I don't know how to invite you guys. How should I invite you? I don't know your names on Facebook. I could search for you on Facebook. Maybe you could send me your Facebook name, and I could send you an invite to that site. I'll do that today, and let's see how that works. All right. Whew. All right, guys, I think that's it for today. That is it for today. Tomorrow's lab. Yes, you are supposed to prepare something for tomorrow's lab. Look at the protocol. It tells you how to do it. The supplies are upstairs in room 240. Kimberly, send me a text. I don't have the time to write down names at this point. So send me a text with your Facebook name. That applies to everyone. I have to get ready for my other class, so that's why I don't have the time to write down your Facebook names. But I'll try to search for you. If you get the invite, please accept it. Okay? All right, so I have to go to my other class. I will talk to you guys. Bye.